Hello everyone. So our today's topic is RNA secondary structure prediction. My name is Hassan Tariq and I am your tutor in this course of computational biology. So basically, there are many keywords, important keywords in this particular topic. First is um, first one is the RNA, and second is structure, and then the prediction. So as we will move forward towards our lecture, we will see what does each of these mean and how what is the combined objective of doing all this. So let's move forward. And first of all, why a brief background or motivation, you can say. So why RNA? RNA is one of the three major types of biological macromolecules. So understanding the structures of RNA provides insights into the functions of this class of molecules. So the detailed structure information about RNA has significant impact on understanding the mechanism of a vast array of cellular processes such as gene expression, viral infection, and immunity. So that is why we want to do this. So RNA, as we know that these are the biological macromolecules, and they provide useful insight into the in understanding what's going on within the cell and in various processes. And it has been involved in why it's important to know its structure, because it is involved in viral infections. It is linked to the gene expression and the cellular immunity as well. So so that's why it is important. So RNA structures can be experimentally determined using X-ray crystallography and NMR. These are the two popular techniques that we use um, to uh, to do structures to determine the structures in the lab. So, however, these approaches are extremely time-consuming and expensive. Obviously, not everyone can do this, and once one need to have a lot of resources to do this and a lot of expertise as well, because these are expensive techniques, and a minor you can say error can produce false results, and so your effort can be wasted, and a lot of resources can also go wasted. So anyways, not everyone can do X-ray crystallography and NMR techniques. These are very these require a supervised, you can say, mechanism of running the experiments. Anyways, so as a result, computation prediction has become an attractive alternative. So the alternative to these experimental approaches are to go for computational approaches, computational predictions. So this particular sorry for the chapter. This particular topic presents the basics of RNA structures and different algorithms for RNA structure prediction, which with an emphasis on secondary structure prediction. So, so a brief introduction of what we are talking about. So RNA is a carrier of genetic information and exists in three main forms. As we know that it's messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA is responsible for directing protein synthesis. Ribosomal RNA provides structure scaffolding within ribosomes, and transfer RNA serves as a carrier of amino acids for polypeptide synthesis. So this is very basic concept of RNA, just to refresh your knowledge about basic molecular biology and what are RNAs and different types of RNAs. So advances in biochemistry and molecular biology have allowed the discovery of new functions of RNA molecules. For example, RNA has been shown to process catalytic activity and is important for RNA splicing, processing, and editing. So these are some of the other known, you can say, functions of the RNA molecules. So a class of small non-coding RNA molecules termed microRNA, which is also abbreviated as miRNA, have recently been identified to regulate gene expression through interaction with messenger RNA molecules. So these are non-coding RNA molecules, but they assist the messenger RNA molecules to regulate the gene expression. So if we compare the DNA and RNA structure, so how it's like. So unlike DNA, which is mainly double-stranded, RNA is single-stranded. Although an RNA molecule can self-hybridize at certain regions to form partial double-stranded structures. So this is one of the major when we talk about the molecules from structure point of view. So this is one of the major differences. That DNA is double-stranded molecule and RNA is a single-stranded. Although it can self-hybridize, and it will show that okay there are some pairing, and it seems to be like it is double-stranded, but it is always single-stranded. Generally, messenger RNA is more or less linear, non-structured, whereas the ribosomal RNA and transfer RNA can only function by forming particular secondary and tertiary structures. So messenger RNA, it is very clear that messenger RNA is a more or less normally it's linear and non-structured, whereas ribosomal RNA and transfer RNA they have you can say secondary and tertiary structures. Therefore, the knowledge of these structures of these molecules is particularly important for understanding their function. So, when we particularly talk about the structure of RNA, um, it can be described at three different levels actually: the primary structure, secondary, and tertiary structure. So, primary structure, like in most of molecules, is the linear sequence of RNA consisting of bases adenine, cytosine, guanine, and uracil. And the secondary structure refers to the planar representation that contains base paired regions among single stranded regions. So as we know that RNA is a single stranded molecule, so it self hybridizes with each other, and this self hybridization is carried out by base pairing um, between different bases, and this forms the secondary structure of the RNA. The base pairing is mainly composed of traditional Watson-Crick base pairing, which is between A and U and G and C. In addition to canonical base pairing, there often exist non-canonical base pairing such as G and U base pairing as well. So the GU base pairing is less stable normally, and it occurs within a double-stranded helix surrounded by Watson-Crick base pair. Finally, the tertiary structure is the three-dimensional arrangement of bases of the RNA molecules. So this is more complex than secondary structure. Here is a pictorial representation of three structures of the RNA. Primary, as you can see, it's the linear representation. Secondary structure, we have some base pairing, which forms a structure. There are loops, there are different elements actually. So we will see what they are called actually. But the main point is that it is a single stranded RNA which has self hybridized, and due to certain um, attractive forces between different bases, and that's why it got this shape actually. And the third one is the tertiary structure, which is three-dimensional structure, which is more complex. So because the RNA tertiary structure is very difficult to predict, attention has been mainly focused on secondary structure prediction. And based on the arrangement of helical base pairing in secondary structure, four main subtypes of secondary structures can be identified, which are hairpin loops, bulge loops, interior loops, and multi-branch loops. So, some main attention has been people has mostly focused on secondary structure because it is relatively easy to work on it. There are less problems to solve while writing programs for these kind of structures actually. So because we want to do this, we want to predict the structure of the RNA through computational approaches. So that's why we normally focus on secondary structure of RNA. So, what is the hairpin? So, different elements of the you can say secondary structure. So, hairpin loop refers to the structure with the two ends of a single standard region connecting a base pair region. I will show you this in diagram. The bulge loop refers to a single standard region connecting two adjacent base pair segments so that it bubbles out in the middle of a double helix on one side. The interior loop refers to two single standard regions on opposite stands connecting two adjacent base pair segments. It can be said to bubble out on both sides in the middle of a double helical segment. And the multi-branch loop, also called helical junction, refers to a loop that brings three or more base pair segments in close vicinity, forming a multifurcated structure. So this is the example of each of them. So you can see how these are, as you can see, this multi-branch loop that pop out of from the you can say there are two base pair segments on the two helical segments on the both sides, and there is bulge loop which is a small pimple like um, structure that pop out in between the two helical structures, and so you can see from these. So there can be additional structural elements as well, like a pseudonode loop, which refers to the base pairing formed between loop residues within a hairpin loop and residues outside the hairpin loop. A kissing hairpin refers to the hydrogen bonded interaction formed between loop residues of two hairpin structures. So this is the interaction between the loop residues of two hairpin structures. Two hairpin structures have their loop residues try to interact with each other. So the hairpin bulge contact refers to the interaction between loop residues of a hairpin loop and a bulge loop. This type of interaction forms supersecondary structures, which are relatively rare in real structures and thus are ignored by most conventional prediction algorithms. So, normally when we talk about RNA secondary structure predictions, we normally focus on these kinds of you can say structure elements. So these additional structure, super secondary structure elements. This is the example of some of them. So pseudonode, kissing hairpins, and hairpin bulge contact. So and now we will move towards the RNA secondary structure prediction methods. Normally, there are two kinds of methods that are being followed in the community, and first one is ab initio method, second one is comparative approach. So now we'll see one by one that how both of them work, and what are what are the cons and cons of both of these. So first one is ab initio approach. This approach makes structure prediction based on a single RNA sequence. The rationale behind this method is that the structure of an RNA molecule is solely determined by its sequence. Thus, algorithm can be designed to search for a stable RNA structure with the lowest free energy. So basically, we this as we see that this as we have discussed that this is the example of computational approaches to predict the structure of the RNA secondary structure. So we look for um, that how the free energy of the molecule can be lowered, and we design the algorithm in such a way. Then on the base of that, we predict the RNA structure. Generally, when a base pairing is formed, the energy of the molecule is lowered because of the attractive interaction between the two strands. So that is the reason that why we go for the lower base pairing, the lower free energy, because the more number of interaction or base pairing in the molecule, the more uh, lower is the energy of the overall energy of the molecule. So that means we design algorithms in such a way that they tend to find maximum base pairing in the structure. So this is the key point here. So, thus, to search for a more stable structure, ab initio programs are designed to search for a structure with the maximum number of base pairs. As I said, we look for the maximum number of base pairs. The structure. Uh, one point to consider here is that we our algorithm can or cannot predict more than one structure, but among them we always choose for go for the structure which gives us the lowest the maximum number of base pairs, because it gives us the lowest free energy. So how do we normally calculate the free energy? Free energy can be calculated based on parameters empirically derived for small molecules. For example, GC base pairs are more stable than AU base pairs, which are more stable than GU base pairs. So it is also known that base pair formation is not an independent event. The energy necessary to form individual base pairs is influenced by adjacent base pairs through helical stacking forces. And this is known as cooperativity in helix formation. So this is the concept of cooperativity. So the main idea here is that if multiple base pairs are present in a structure and they are adjacent to each other, so they influence each other, actually the energy of the RNA molecule. So this is called as cooperativity in helix formation. If a base pair is next to other base pair, the base pair tend to stabilize each other through attractive stacking interactions between aromatic rings of the base pair. So that's what I already described. So this is the concept of cooperativity. As I already mentioned, that we look for the region where we can get maximum base pairs, and because this makes the structure of the RNA more stable, and the energy of the molecule of the RNA is is lowered is lowest. If the base pair is adjacent to the loops is adjacent to the loops or bulges, the neighboring loops and bulges tend to destabilize the base pair formation. So if there is a loop nearby or any bulge nearby, so it also tends to destabilize the base pair formation. Ab initio method works by first finding all possible base pairs patterns from a sequence and then calculating the total energy of the potential second structure by taking into account all the adjacent stabilizing and destabilizing forces. So this is a very key point here, that how the overall approach of ab initio method works. We first find the possible base pairing in this sequence, and then we calculate the total energy of the second structure, um, and take into account the what forces are can be there that can stabilize or destabilize the structure. If there are multiple alternative secondary structures, the method finds the conformation with the lowest energy, meaning that it is energetically most favorable. So, so in searching for the lowest energy form, all possible base pair patterns have to be examined. So there are several methods for finding all possible base paired regions from a given nucleic acid sequence. So these are the three approaches that normally considered to be common one and a popular one: dot matrix, dynamic programming, and partition function. So uh, the dot plot method or dot matrix is effective in finding a single secondary structure in a small molecule. So if your molecule is small and so it is very effective in finding a single secondary structure. However, if the molecule is large and it contains multiple secondary structure segments, um, choosing a combination that is energetically more stable among a number of possibilities can be done by dynamic programming. So, dynamic programming is used in this scenario where the molecule of the RNA is large and there are multiple possibilities of a secondary structure. So, we have to choose which one can be more stable. So, the dynamic programming is best in this particular scenario. But dynamic programming method produces one structure with a single best score. However, this is potentially a drawback of this approach because in reality an RNA may exist in multiple alternative forms with near minimum energy but not necessarily the one with the maximum base pairs. So multiple alternative forms of us same RNA residue can be present in reality, which have almost similar energy, um, but their base pairing cannot be similar or cannot be maximum. So that's why dynamic programming is good approach, but it has this drawback that it cannot tend to this problem. It cannot solve this problem. So the problem of dynamic programming to select one single structure can be complemented by adding a probability distribution function known as partition function. So we can add a partition function. So to select one single structure, um, so this calculates a mathematical distribution of probable base pairs in a thermodynamic equilibrium. So it calculates actually mathematical distribution thing, okay, and of what of probable or in probable here means the base pair which are likely to be which there and present there in a thermodynamic equilibrium. This function helps to select a number of suboptimal structure within a certain energy range. The following list two well-known programs using the ab initio prediction method. One is mfold, other is RNA fold. So, and Nussinov RNA folding algorithm is an example of ab initio method, and this has been a very common one, uh, and it has been here for some time now. Um, so Nussinov algorithm is a basically dynamic programming approach actually. So algorithm is a popular example of a class of algorithm known as dynamic programming algorithms. The main idea behind these algorithms is that we can break down the problem into many subproblems, which are easier to compute than the full problem. So once we have obtained the solution for the subproblems, we can retrieve the solution to the full problem by doing something called a backtrace. So this is the example of how dynamic programming works. So, um, uh, Nussinov folding algorithm. The objective is to obtain the optimal pairing on a string of nucleotides. In order to know how good our structure is, we assign a score to it. One possible scoring scheme could be adding 1 to the score per paired set of nucleotides and 0 otherwise. If the nucleotides are going to be paired, so we will score it 1 and otherwise 0. So the key here is to remember that the RNA structure, RNA molecule is a single stranded, and the base pairing here is done between different residues of the RNA. So the key here is to remember this, and so the scoring scheme is unlike the DNA structure. So in other words, we want a pairing that will give us the highest possible score. We can write this quantity as opt. Opt is optimized, where I and JJ are the indices. Sorry, there should be one IJ are the indices of the sequence between which we obtain the pairing score. So IJ here are the positions on the residue between which pairing is determined or is predicted. After we have a way of measuring how good the structure is, we need a way to evaluate scores given a subsequence. To do this, we set some rules on the structure of an RNA sequence. If IJ form a pair, the pair I and J must form a valid Watson-Crick pair. So that should be a valid, like A bind with U and G bind with C. And also I less than J minus 4. This ensures that bonding is not happening between positions that are too close to each other, which would produce steric clashes. So it should be the rule here is that the two positions on the single strand of RNA, they should be 4 bases apart at least, so that they can bond with each other. Otherwise, the bonding will be unstable if they are too close, and this will not the structure will not be stable, or it will not lead to, you can say, good prediction. If pair IJ, KN are in the structure, if these are the two different indices, IJ and KL are in the structure, these are the two pairs, you can say, then I less than KJ less than L. So this ensures that there is no crossing over of pairs, which would result in pseudonodes. So what does that mean actually? So imagine a single stranded RNA. So there are two base pair I and J. So the point here is that they should not be in each other's way, so that when the bonding is formed, is predicted, the bonding is crossing over each other. So that means in actual the bonding cannot be formed if they are in the way of each other. So that's why we have to follow this rule. So no base pairs in more than one pair. No base appears in more than one pair. So how it works? Create a matrix with RNA sequence on top and right. Set diagonal and node tridiagonal to 0. Start filling each empty position in matrix by choosing the maximum of 4 scores. So let's see how. So that's how. And this is a single stranded RNA, and we are in Nussinov algorithm. We write it in the horizontal and vertical axis, just like this, and fill the diagonal with 0 here. As you can see, there is 0 on both of the diagonals. So then this is I and this is J. This is the same RNA molecule which is written here and here. So we are just want to fill this now. So what we do here is that we have filled the diagonal zeros, and now we will fill it. We will fill this position, like position here 2, which is under G, with the maximum of the values here. The key here is to remember that if G and G, this can be paired, just add 1, and also add the maximum of the 3 here. So as we can see, there is no the G and G cannot be cannot be base paired. So uh, so the base pairing score is 0, and the maximum is also 0 because left sorry bottom, left, and diagonal, both all three of them have zeros. So no, there is no maximum value, so the 0 will be there. So and so on, we will move forward and fill the whole table. So this is the how we fill the, so Si plus 1 and J. So we will take the maximum of whatever possible here. Either one of them will be maximum, so whatever the maximum is, we will fill that position with that. So I plus 1 and J means, as we can see, so I here is the vertical column. So if for example I am taking this, so I plus 1 means the lowest one, and J remains the same. So so is the then I j minus 1 is the left the previous element to that position, and this one is the diagonal one, and we also add energy of the pairing, and this is the left row and bottom. This is the left row and summation of left row and bottom column. um, um, how we calculate it, I will try to explain it, that if there is if there are values there, for example, if I am talking about this particular column, 6 a6 and a5 1, this one, so we will see that how can be it can be done, that either three of them have maximum value. If not, then we will go for the diagonal. How we go for the no sorry diagonal, it's the you can say left row and bottom column. So this is the bottom column and this is the left row. So whatever we sum these values and whatever the maximum, we put it here. But in this case it is 0, so here it will be 0. So this is a general idea of Nussinov's algorithm. Obtain an RNA sequence, create a scoring matrix, and place sequence. Set the diagonals, devise scoring scheme, and if set each matrix position by calculating 4 conditions, which I mentioned. These are the 4 conditions. Set the maximum value from the 4 condition into the matrix position. If this is the last position, then trace back the element. So and you will get the you can say what the RNA structure is, what base pairs are possible there. The advantage of Nussinov's algorithm is show patterns of RNA structure prediction by dynamic programming. Only one solution problem can be overcome. Because there isn't only one outcome of this, and prediction of crossing structures can be seen as extension. If we want to see the two base pairs are crossing each other, so we can extend this algorithm to solve this as well. So limitations of this: base pair maximization does not yield biological relevant structures. So how not relevant structure, no stacking of base pairs considered, loop sizes are not distinguished, no special scoring of multi loops. So only one structure predicted, so base pairing maximization cannot differentiate structures sufficiently well, possibly many optima, no suboptimal solutions. So crossing structures cannot be predicted. So these are the 3 limitations of this. So the other approach of RNA secondary structure prediction is comparative approach. Comparative approach uses multiple evolutionary related RNA sequences to infer a consensus structure. This approach is based on the assumption that RNA sequences that deemed to be homologous fold into the same secondary structure. By comparing related RNA sequences, an evolutionary conserved secondary structure can be derived. So that's how it works. To distinguish the conserved secondary structure among multiple related RNA sequences, a concept of covariation is used. It is known that RNA functional motifs are structurally conserved. To maintain the secondary structures while homologous sequences evolve, a mutation occurring in one position that is responsible for base pairing should be compensated for by a mutation in the corresponding base pairing position. So, to maintain base pairing and the stability of secondary structure. So, this is the concept of covariation. If there is a mutation at one position in the base pair, then there should be a mutation in the relevant base pairing as well. So, this is covariation because these are functionally conserved. RNA functional motifs are uh, structurally conserved regions. So any lack of covariation can be deleterious to the RNA structure and function. Based on this rule, algorithms can be written to search for the covariation patterns after a set of homologous RNA sequences are properly aligned. The detected correlated substitutions help to determine conserved base pairing in a secondary structure. Comparative base algorithms can be further divided into two categories based on the type of input data. One requires a predefined alignment, and another does not. The algorithm that use pre-alignment requires the user to provide a pairwise or multiple alignment as input. So these alignments can be obtained by using standard alignment programs like T-Coffee, Cluster. And based on the alignment input, the prediction programs compute the structurally consistent mutational patterns such as covariation and derive a consensus structure common for all sequences. In practice, the consensus structure prediction is often combined with thermodynamic calculations to improve accuracy. So, so in practice, consensus structure prediction they are normally combined with the thermodynamic calculation, which is kind of ab initio method to improve the accuracy of the structure prediction. This type of program is relatively successful for reasonably conserved sequences. The requirement for using this type of program is an appropriate set of homologous sequences that have to be similar enough to allow accurate alignment, but divergent enough to allow covariation to be detected. So they should be similar, but they should be divergent as well, so that the concept of covariation can be applied. If this condition is not met, correct structures cannot be inferred. So example of such program is RNA unfold. So second is that the algorithms which you do not use pre-alignment. So these type of algorithms simultaneously align multiple input sequences and infer a consensus structure. The alignment is produced using dynamic programming with a scoring scheme that incorporates sequence similarity as well as energy terms. Because the full dynamic programming for multiple alignment is computationally too demanding, currently available programs limit the input to only two sequences because the dynamic programming is an algorithm which is computationally too expensive. So such the example of such program is fold align and dynalign. So this is the end of the today's lecture. So hope to see you again with the next topic next week. So if you have any question, we will get to that after this in a special session. Thank you for